So in today's podcast, it's my pleasure to talk with Maya Opendak. She's an assistant professor of neuroscience at Johns Hopkins University School of Medicine and also in the Kennedy Krieger Institute. And maybe she can talk a little about that institute because it's very relevant to her work, which is aimed at understanding the developmental neurobiology of social behavior and the impact of early life experience on infant brain development, in particular looking at specific neural circuits in the brain of developing infants. And a lot of the work she does, it's impossible to do in humans, and to understand what's really going on in the brain, say with early life trauma or deprivation, which can also be a thing infants experience early in life. She's getting into the nitty-gritty of what's happening in certain neural networks. She'll talk about that. So Maya, welcome.
Thank you, Mark. It's really exciting to be here.
And first, I'll ask you about kind of your background. When did you become interested in early life development and brain development in particular?
So I was doing my PhD in the lab of Elizabeth Gould at Princeton, and I was for years studying dominance hierarchies in rats. And I was studying social memory and how adult-born neurons in the hippocampus respond to social disruption of dominance hierarchies, and sort of the encoding of that hierarchy information and the role of adult-born neurons in that process. And we found that if we disrupted a hierarchy by replacing the dominance between two hierarchies of rats, you'd have this prolonged fighting once these new animals were there. So normally when hierarchies form, fighting decreases and sort of adaptive in that way, but we kept the fighting going by replacing these dominants. Their position went way down. Adult neurogenesis went down in all the animals in the disrupted hierarchies. And we also found that the animals that had been in disrupted hierarchies showed a change in their social behavior weeks later. And that was, controlled by neurons that were born during the disruption. So if we knocked out those neurons, we didn't see the change in their social behavior. So we could see that even this brief period of environmental disruption in adulthood could remodel social behavior through these new neurons. And I ended up going to some talks near the end of my PhD about early life stress and outcomes of resource deprivation and scarcity in humans, and how that related to educational outcomes and stress and socioaffective issues. And I just thought, okay, I'm already really interested in how the environment is sculpting the brain to change behavior. And it makes sense to go to a time when these circuits are actually being built to ask, how does the environment sculpt the long-term programming of social behavior in this very vulnerable period of early life? And
you're looking, you're focusing on the infant brain, but what's important, can be very important, is actually the environment and experiences of the caregiver.
Absolutely. Yeah. So actually our work shows that really the most important thing for the infant is what their caregiver is experiencing, because that's what's communicated to the infant. So we study infants, but the way we disrupt their environment really is to disrupt how they process the caregiver. And one way to do that is to give the caregiver, so in this case, we take a female rat that just gave birth to pups, and we take away part of her bedding resources. And many labs do this. This limited bedding and nesting models are pervasive in the literature. This specific model that we use is called the scarcity adversity model, which is slightly milder than some of the others. As a result of this resource deprivation, the mother rat is highly stressed, and she roughly handles the pups much more. That can include carrying them in her mouth or stepping on them. And as a result, so 5 days of this chronic experience when pups are in a sensitive window, which has been identified with lots of research, the postnatal day 8 to 12, for example. Having that experience with this stressed mother leads to increases in stress hormones in the pups, and also, immediate and lasting structural and functional changes in how they respond to, how they process fearful cues, how they process social information. And so that's kind of the subject of my work now, is to ask, how does this experience with the mom, how does this early experience change how they actually process the maternal cue?
So in that animal model, then, so that, we're really not supposed to do this as scientists, but I think it's actually very useful to kind of put yourself in the position of the mother, right? So a nest is kind of protecting the babies. They're all together in a physical location, and they have this physical nest around them. And that's a good situation for them. It's a good situation for the mom because she knows where they're at. She knows they're semi-protected, right? And so when you remove the nest or the bedding and the amount of it, then she's worried probably that something bad's going to happen to her developing child. Does that make sense?
Yeah, I think that makes sense. We see that she's with this small amount of bedding, she like constantly is trying to gather it like over and over and over again. And so it's just, and what's interesting in this model though, so she's spending a lot of time building and rebuilding this nest, or trying to build a normal nest. We see something kind of interesting is that sometimes if the mother rat has already been exposed to this in the past, she'll make a nest out of food. So there's like an adaptation that goes on, which is really interesting, and we should definitely study it.
Huh.
And I mean, maybe some people are already studying it, but I'm interested in studying it as well. And so that you see this adaptation. But what I guess what I wanted to also say is that there's this increase in rough handling of pups, but she also still nurses them like the same amount in this model. And she still grooms them. So nurturing behaviors are still happening in addition to this repeated nest building and rough handling of pups. So it is, and one of the really interesting things we've observed from this model. So if we actually, the one thing we really can't do in children, and rightfully so, is we can't measure what's actually going on during the early life trauma, right? Well, we can do that in rats. And we implanted wireless telemetry recording devices subcutaneously that were recording from the cortex in young pups during this 5-day period. And we could see, these were brief recordings, but we measured what was the cortical response, cortical local field potentials, oscillations, brain waves, what was their response to
So these are, Maya, sorry for interrupting. I think I'll hold my finger up when I have something.
Okay.
So is this like EEG recording, electroencephalogram type recording?
Yes.
Yeah. And these people,
but it's actually in the brain. The wires actually go into the brain.
Yeah. And people may not appreciate these pups are very small, and
Yes.
and the surgery is probably not trivial.
Certainly not. This surgery in particular, because the probe is kind of large. I mean, the implant, and the really difficult thing with pup surgeries is to make sure that the mother still accepts them and treats them normally. And that means that they have to be fully recovered and indistinguishable from non-surgerized pups. Wow. So that kind of adds a whole extra texture to things.
Wow, yeah. Then again, thinking about in the human setting, so I was thinking as you're describing this model, reducing the amount of nesting and so on, I'm thinking of, say, a single mom who has several children, and she's working, right? And so she's worried about her her kids and whether she can provide enough resources and so on. And is this the kind of thing that's trying to be modeled here? You know, ultimately what you want these models to be relevant to, like, real world human. Is that reasonable?
Yeah, I think we have to be careful because we don't want to say that poverty causes child abuse, right? So that's not what we're saying. But I think what we are saying is that what we see in a lot of this work is that the best way to help children is to help the mother. So if there's
but that's what I was getting at, like the kind of increased stress on the mother is it doesn't have to be a poverty situation, just whatever, you know, increased stress, you know, maybe stress between a spouse that kind of secondarily affects the developing child.
Definitely. And I mean, this study using the LFPs, which are an analog of EEG, what it showed was that, so these pups, like I said, even when the mom is stressed, she's still nurturing them. And normally these nurturing things like milk or being groomed produce this expected change in oscillations in pups. And that's been shown in the past. But if those exact same nurturing behaviors are done when the mom is stressed, they don't change the pup's brain in the expected way. So, which is really interesting because and that actually is, you know, there is a direct parallel to that in the human literature, showing that the quality of attachment between the caregiver and the child influence the degree to which the caregiver can influence the child's brain in EEG work. So this is like a really important finding to us because it shows that the actual salience, I guess, of caregiver changes when the caregiver is stressed.
Yeah. And I guess before I go on with the questions I have for you, I just viewers and listeners might want to know that there's several other podcasts I've already done on human child development. And one was with Chuck Nelson from Harvard on childhood adversity, and he like studied children in Bangladesh and some other places, really interesting work. And then I did recently one on with James Blair, who studies like childhood, essentially conduct disorder in children and kind of antisocial behavior. And, you know, kind of there's this critical, and he pointed out there's this critical time window during development of children that there can be interventions, behavioral interventions that can help them a lot. But there seems to be this critical window. If they reach adulthood, you know, and they have conduct order, they have a really high probability of going on to have like a psychopathic behavior. But anyway, so the connection with your work becomes fairly obvious in what you're talking about now, getting down to the circuits. So you talked about recording EEG, but what about specific neuronal circuits in the brain that might be altered and contribute to like a long-lasting problems with social interactions? Oh, and in that regard, I also did a podcast with Matt State on autism, which kind of one of the manifestations of many instances of autism is kind of being withdrawn and not want to have social interactions. Okay, so anyway.
No. So I'm glad you asked. So we've looked at a lot of different brain circuits, or several different brain areas, I guess I should say. And a lot of the work sort of starts with the medial prefrontal cortex, amygdala, and hippocampus circuit, because these are really affected in PTSD and, you know, especially with early life trauma. And I'll start with the amygdala. So first, there's structural changes that occur in the amygdala after this chronic early stress. But like, for example, the volume of the amygdala can change. The amygdala is an important brain region for learning about fear, but it's also important for other things, including social behavior. So what we've observed is that after this adversity, this early life stress, there's this decreased social behavior with the caregiver and also throughout the lifespan with peers. And that is caused by amygdala hyperactivity during the social behavior. So if you can look at amygdala activity during the behavior and see that it's upregulated. Mm-hmm. And if you decrease that hyperactivity with optogenetics in pups, for example, you can prevent the emergence of the social behavior deficit. Oh, no, you can rescue the social behavior deficit. And you can take a normally reared, control reared animal, stimulate amygdala neurons. So these are principal neurons in the basolateral amygdala, and you can inhibit social approach behavior. And those changes are persistent. So even when they're 22 days old and you do this exact same manipulation, you can suppress the amygdala hyperactivity, rescue social behavior with peers. You can create an adversity-like phenotype by stimulating the amygdala. So with that information in hand, so we adapted optogenetics for pups to ask this question in like a cell type specific, temporally sensitive way. But that work had also been done using mucimol. So we wanted to build on that. So we added, the pharmacological approach of.
So that's an acetylcholine receptor agonist.
Yeah. Yes. So the benefit of using optogenetics would also be to do a specific circuit approach. And so what we did next was we saw that normally dopamine signaling is important for plasticity in the basolateral amygdala. And it's been shown in adults that fear learning depends on dopamine signaling in the basolateral amygdala. And we observed that these animals that had been experienced adversity with the caregiver, and I can get into a little bit more of how we specifically can study that. We can also study adversity alone versus social adversity. But what we saw was that whereas typically the caregiver suppresses dopamine release in the basolateral amygdala, repeated adversity with the caregiver results in the opposite. So there was this increase in dopamine in the basolateral amygdala in the presence of the caregiver. And so if this is a brain region that's important for fear learning, we thought, okay, well, that seems like something you don't want to have going on. That seems that there would be plasticity going on in this brain region important for fear. They're not supposed to be experiencing fear in the caregiver's presence. So we did some correlational studies looking at, activity of the ventral tegmental area, which is the source of dopamine to the basolateral amygdala. We saw that it was the mother's presence failed to suppress that brain region following adversity rearing. We measured dopamine directly using HPLC and microdialysis and mass spec. That was work done by colleagues. So then we went back to the So I did a pharmacological dopamine inhibition during the adversity itself. So I just blocked dopamine receptors in the basolateral amygdala with a drug, and pups were experiencing this social adversity. And we were able to prevent the emergence of the social deficit.
So probably most listeners and viewers, when they think of dopamine, they think it's like a rewarding thing that like it happens when you're experiencing something pleasant. But you're seeing, so this has to do with specific circuitry, because you're seeing an increase in dopamine specifically at these certain synapses on certain neurons in the amygdala has kind of the opposite.
Yeah.
Is that right?
Yeah, I think that the role of dopamine is really projection specific. And if we were looking in the nucleus accumbens, which is what my lab will be doing soon, and is in the process of doing these experiments, you might see a different outcome. Yeah. Nucleus accumbens I think is kind of more frequently associated with reward than the basolateral amygdala. Dopamine projections to that brain region kind of have a different, they're more important again for fear learning. So after we did this pharmacological approach, which was initially like just circulating dopamine, circulating the drug in the bloodstream. So we did a systemic approach, but then we did a central administration of a dopamine antagonist during adversity. And then we went back to optogenetics and we used, transgenic animals to specifically target projections from the ventral tegmentum to the BLA.
Well, which gene manipulated in the transgenic?
It was a TH-CRE rat. So tyrosine hydroxylase expressing neurons.
So it's the enzyme critical for production of dopamine.
Yes.
Yeah. Okay.
So neurons that had this tyrosine hydroxylase expressed CRE recombinase, and then we did a CRE-dependent viral approach. So anyway, we targeted this specific circuit, and it was also, it produced a really similar outcome to just controlling the basolateral amygdala, which was that we could rescue social behavior after adversity, and we could also disrupt social behavior by stimulating this circuit.
And is this all with this deprivation model with the nesting, or you use another model too, this deconstructed adversity model?
Yes. So the scarcity adversity model is really great because it's sort of kind of easier to do. You just, it's high throughput, it's naturalistic, but there's so many variables that are changing in the animal's environment, right? We wanted to understand specifically what is the role of the mother being the source of stress or associated with the stress versus them just having some repeated aversive experience. Right. So we took pups, and they were either shocked, they received mild shocks alone or with the mom every day for that same 5-day period. And so we could separate, what happens to the brain and behavior when they're just stressed, because it's been shown in children as well that different types of stress, like, living in a war-torn area or versus child abuse and neglect, they produce different outcomes. And so we kind of were able to model that a little bit.
So the animals are the pups in the cage with the mom, and you're selectively shocking the pup, and the mom is observing the no, okay.
No, sorry. I just wanted before, I didn't want to interrupt, but so we take the pups out and we put them with an anesthetized mom that to them is indistinguishable because they can't see or hear. And so they are guided by odor. So she has the same odor as their biological mother. And she's anesthetized to prevent her actual interference in the experiment. So we just have her odor there.
I see.
And they receive these mild tail shocks. And they're most of the time in the beginning of the experiment, they're still attached to her ventrum, which is their normal behavior. There's like nursing or sleeping. So, but they're receiving these mild tail shocks, which is kind of trying to replicate the rough handling that they experience in the nest, the naturalistic nest environment. And you can also do this with shocks, but also with increasing their stress hormone levels and exogenously with like corticosterone injections. So you can create, so I mentioned amygdala dysfunction or hyperactivity, and I mentioned social behavior deficits. So if you just stress these animals repeatedly by injecting them with cord or giving them shocks, you see hippocampus changes. And that's really consistent with all the work by Bruce McEwen and Sapolsky and everybody. But it has to be in a social context, even if the mom is anesthetized.
I see.
Then you get amygdala deficits or just changes in structure and function and social behavior deficits.
And so that's really interesting. So the, activating the olfactory pathways, for the sense from the mother is like a critical factor.
That's the primary sensory modality that's sort of controlling this process.
Really interesting. And okay, so you talked about the amygdala, right?
Oh,
And what about other brain regions? You know, I see one that you're looked at, which I don't know much about actually, is the habenula.
oh yeah.
And that region hasn't been talked about in any of my podcasts before. So could you
Yes.
describe
Oh, I'm
this
the
mysterious brain region?
So it's actually, so that's been the subject of, now that I have my independent lab, we've been really focusing on the lateral habenula, which is this brain region, which is sort of highly conserved across species. It's the interface between the forebrain and monoaminergic systems. So like the, so
Serotonin, norepinephrine
yes,
in the brainstem.
yeah, and dopamine as well. So
And,
the lateral habenula, so we had this information about, I mentioned cortical oscillations and I mentioned dopaminergic release from the VTA. And so the habenula is kind of in between those
okay.
circuits. And the habenula is important for behavioral flexibility. And it's been shown to be, it's one of these brain regions in animal models of depression. It's really, really across all these animal models of depression in adults. It's consistently shown to be hyperactive in all of these regions. And this brain region, one thing that it does is that it sends a projection that ends up inhibiting dopamine release from the VTA.
And
So
these neurons in the habenula, well, this is true throughout the brain essentially, except for a few exceptions. They use glutamate as their neurotransmitter, excitatory transmitter.
yes, like
Okay.
over 97% of neurons in the habenula are excitatory, but they project to, one of their projections is the rostromedial tegmentum, which sends a GABAergic inhibitory projection to the VTA.
So they activate inhibitory
yeah,
neurons. Yeah. Okay.
and it's interesting because so this brain region, it's really important for behavioral flexibility. So switching tasks as information levels change. It's robustly active in response to aversive cues. It's considered like the anti-reward brain region. So in the absence of expected rewards or something aversive like shocks, habenula in adults is robustly activated. So, I mean, it has different projections, but that's one of its main sort of canonical roles. And there's really not that much known about what it does in early life. There's some evidence in humans that in autism spectrum disorder, the habenula of children is enlarged. And so we thought that this might be a really fruitful place to look for, as a site of plasticity in early life that, again, could help us synthesize these different types of information that we've been seeing. And so what we've seen is actually really interesting. We're doing like sort of a fundamental characterization of the habenula in development. So starting with just not even task dependent, but volume changes with early life adversity, protein expression in the habenula related to its activity. So we looked at 2 ages. So we looked before weaning and after weaning in pups. So, around 15, 16 days old, and we looked at 10 days later. So they're weaned at 21. And this age is really important because that's when social behavior switches from maternally oriented to peer oriented. So we thought that the habenula might also have a role in this flexibility that's involved in that change. Because again, social behavior in early life is highly biased to be pro-social. And that's adaptive. But as you get older, like the social behavior has to involve more decision making. And so we thought the habenula, we could sort of track that normally, but also and see how that changes with adversity.
So it's so the prefrontal cortex is important in decision making. Actually, I had a podcast on that. And then the habenula sits between that and, okay, I was going to ask you, so early on in our conversation, you mentioned your early work in the hippocampus and the kind of in neurogenesis that kind of got you interested eventually in it. So I expect there must be connections between the habenula and the hippocampus. or not?
I would check that, actually. I don't remember. I mean, that's not, let's see.
Because all these things, at some point, there has to be some learning and memory component, and
I mean, I think yeah, I'm not
and I
actually
think, you
sure.
know, I should tell you, so I know Liz Gould because I had a graduate student, Alexis Stranahan, who got
Yes.
a PhD with Liz, but actually she did her PhD dissertation work in my laboratory at NIA on effects of diabetes and obesity on hippocampal plasticity and so on. And also in my an investigator, independent investigator in my laboratory. So the word laboratory at NIH, it's kind of like the equivalent of a department at a university. And I was a chief of the lab, which was like a department head. Anyway, was Henriette von Prague, right, who discovered that exercise stimulates neurogenesis. And then Henriette, when she was in my lab, she did all this nice like track tracing with rabies viruses and stuff, and seeing what are the inputs to the newly generated neurons that incorporate into the dentate gyrus. And I'd have to go back. My recollection, maybe that's the one place I heard the word habenula before. So I think maybe there are connections.
I mean, I know that the habenula, I mean, it's right next to the hippocampus, and all the oscillations in the habenula, they've shown that those are from the hippocampus.
The
But in terms of,
theta rhythm.
yes.
Yeah, yeah.
And so I don't know about direct connections. I mean, the habenula does not have a direct connection with the amygdala, for example, which is really interesting to me, considering that I think that they do sort of similar things, but that's very simplistic. So I'd have to see. I mean, it's not one of its primary connections.
Well, and I expect
hippocampus.
this hasn't been looked at much in development at all, I don't think. So you're at the forefront of all this stuff. So
Yes,
maybe a few years from now you'll be able.
yeah, I think, and yeah, that's exactly true. That, and also like the hippocampus is late developing in, it's not really, at least it's not participating in the social behavior, for example, in these really young pups that we're looking at. Yeah, I don't know of a direct connection with the hippocampus, but it is adjacent to the hippocampus, so I'd be surprised if there was nothing there. But what we saw was that if we just targeted the habenula with something like, so initially we used DREADS, like a chemogenetic approach, which has the advantage over optogenetics that it doesn't require the implanting of fiber optics, and you kind of have a longer term manipulation.
So you use kind of go to the technical stuff. So you're using virus?
Yes.
To infect only certain cells where you put the virus, and then the virus has genes encoding ion channels that are sensitive to a drug.
Yes.
Right. So kind of the bottom line is you can give a drug systemically, and it's going to only affect the activity of neurons in a specific region of interest.
Yes.
Okay. All right.
Yeah, that's the idea. So Liz Gould, for example, actually has done DREADS in pups. So a student in her lab published a paper where they injected 3-day-old pups with this virus. And then they did a manipulation. I think it was at 15, maybe a little bit younger. So we did a similar thing. So we expressed this inhibitory DREAD, which was a modified human muscarinic receptor.
Oh.
And it's kind of a nice, like, less invasive approach, which is good for pups, because like every single manipulation you do, again, the bottleneck is, is the mom going to rip the implant out and like things like that. And also optogenetics, I really appreciate, though, now having been doing DREADS for the last year and a half, like you don't need as many animals, I think, in opto, because you can do within subject comparisons. You just have the light on versus off. And you can do within subject comparisons with DREADS too, but you have to do the test twice. So you need to have like saline controls. You need to have pups that get CNO, the ligand,
And
without.
with epigenetics, you can excite or inhibit the neurons for very defined time periods, whereas you got a drug, the drug stays around a long time. And
Yeah. So what we observe, though, with limitations of dreads aside, with many controls, many animals, we saw something really interesting, which was that. So we had a little bit of correlational data showing that the habenula seems to be. So we looked at 2-deoxyglucose uptake, so metabolism of this radio-labeled glucose was increased in the habenula when there was social information combined with aversive information, which actually most brain regions in pups, looking at the same age, we see the opposite. We see that the presence of social context will decrease especially the mother will decrease, particularly the mother, I should say. That's what the experiments are using. That maternal presence decreases brain regions metabolism of glucose, which is a proxy for regional activation. It's like a PET scan. There's a threat present, like a shock, and the mother is also present. Usually in a typically developing animal, her presence will decrease the response to that aversive cue. But in the habenula, it was the opposite. So we saw that the combination of the social cue and the threat cue increased habenula activity. So we wanted to see what the habenula was actually doing. Like, how did it control behavior? Because in that assay, we didn't look at behavior. We just looked at neural activity in this passive task. So we gave the animals these social behavior tests where, there's the normal social behavior test where they can either approach a pup in a cup or an empty cup, which is the 3-chamber test, very common. And then we added a test where we piped in a threat. So we piped in predator odor. We used fox urine. So that was ambiently there. And then we let them choose how much time they wanted to spend investigating a novel social partner. So it was kind of like, forcing them to make a decision to either, be afraid and not move, and this is a cue that they do respond to in young animals, like the predator odor. They can smell that. So, and it elicits a reaction. So we did these social behavior tests. And what we found, and we also had animals, so we tested them at 2 ages, before and after weaning. So when they're 18 days old and they're 28 days old. And we also looked at adversity reared pups that had this scarcity adversity models. There's many variables, many drug conditions. It was a huge study. And we found that in the younger animals, so I'm going to focus on this test that has the 2 pieces of information, the social and the threat in the air. When you inhibit the habenula, so we started with an inhibitory project, like a loss of function, it decreased approach towards a social partner. So, and then we saw the opposite when they were 28 and they're older, that inhibiting the habenula increased approach to a partner. And so then we did a gain of function study with the excitatory dread, HM3DQ, and we saw the opposite, so we saw basically that in pre-weaning pups, the habenula decreases social approach towards a partner when there's a threat present. But when they're older, it increases social approach. Oh, I'm sorry. I think I'm saying it backwards. When they're younger, pre-weaning, it promotes social approach under threat.
Okay.
And when they're older, it decreases social approach under threat. So which kind of is a transition that I can I can make an intuitive explanation for that, which is like when they're younger, social partners are a source of safety, right? But when they're older, social partners might actually be threats themselves. So it seems like the habenula is controlling that switch.
So I'm thinking about it. Okay, so there's a fox, and so when they get older, they realize, hey, if I run faster than my brother or sister, I'll get away from the fox and I'll survive. And it's kind of like their self-survival type thing when they get older and can kind of think about it more. Is that?
Yeah, I think so.
Okay.
I think especially at the younger age too, like there's a fox and they're like, okay, I'm gonna run to my mom. And that's the habenula is promoting that behavior. And then when they're
Are
older,
they're safer when they're together in the nest
yes.
or that kind of thing?
Yeah, I think of it that way.
Yeah, interesting.
And then again with the another advantage of viral approaches, unlike, just basic pharmacology tools, is that we can look at circuits. And so what we saw, we started looking at, because of other correlational work and work in adults, showing that the medial prefrontal cortex projection to the habenula, which is glutamatergic and it's unidirectional, in adults that inhibits social approach behavior. And we also saw that in these younger animals too. So that was kind of cool. That was not when there was a threat present, but it was sort of interesting. But now the next step is to look at, okay, in terms of the habenula switching, what they do at the 2 ages with threat present, downstream projections from the habenula might tell us what's going on, like what circuits are controlling that process, that transition. And another cool thing that we're doing is, to ask, how is the habenula doing different things at each age in the same test? We're doing experiments where we're tagging neurons that are active during that social behavior test and seeing what is the overlap when they're 10 days older doing that same test. And so far, we don't see any overlap.
And so you mentioned you're starting to look like at structural effects, like at a gross level, the size of the habenula. Have you been looking, counting synapses or dendritic spines, which is kind of a readout of synapses?
Not yet. It's still very, very coarse. We're just starting with volume and not sure yet if there's any effects, but hopefully we'll have some effects of adversity. There's certainly an effect of development where it gets larger between, 18 and 28, but we don't necessarily see yet whether that's the case with adversity.
Oh, okay. And there's another brain region. So in a recent episode, there's a neuroscientist at Harvard, Michael Ferguson, who he's studying the neuroscience of religious beliefs and mysticism. Okay. And so he does functional brain imaging in humans, right? And has them while they're being imaged in the magnet, they're, as religious people, and they're reading like religious texts or whatever. And then he used this brain connectome project data to try to integrate with his functional imaging. And it turns out people highly religious, when they're having these kind of religious experiences, the circuit in the periaqueductal gray seemed to be a focus of everything. And I didn't know much about like that region, just like I didn't know much about the habenula, but turns out that brain region is important in caregiver child, mother-child bonding in early development. It's also involved in fear responses and pain.
Yes.
So that might, altruistic behavior. So that might be another region.
Oh,
for
for sure.
look.
That's super interesting. We've been focused on, especially the manipulation where we shock the pups alone or with the mom. The periaqueductal gray would be a region to look at, and with those animals for sure. And yeah, I mean, we've been thinking of staying, in the nucleus accumbens lateral septum area, because those are the lateral septum is important for social reward. But yeah, we're kind of switching to like a reward mindset now. But PAG, that sounds really interesting. I don't know much about its development.
So it's easy to plan your work in the future and get really great papers, because so you have a PhD student come in or a postdoc and say, okay, what about these circuits?
I know.
What about this?
It's like a lot of
and you get some preliminary data and then put a grant proposal on. Oh, we're gonna look at this.
Yeah. Yeah,
Yeah.
that's the game. No, but like,
Yeah.
but there's a lot of these tools are now in our hands, right? To study these circuits very early and
Yeah.
study them within the same animal sometimes. And so yeah, I mean, it would be good to have like a consistent through line of like some sort of hypothesis, but it is all a lot of it's a black box, with circuits
Yeah.
especially.
Yeah. And then, so are you thinking about going to kind of a different level? Looking at the molecular level or specific genes that are altered in now it's with new technology, it's even possible to look at gene expression and or gene expression patterns in individual neurons, I guess. And then there's this whole field that's very important in development and kind of transgenerational effects, which something we haven't talked about. Like, it's pretty clear now that the environment of mom and pop when they conceive a child can influence that child at different levels. There's a lot of work on obesity or stress. Like, if you stress a mom during pregnancy or something, then you take the pups and you put them with another mom, they still have some behavioral alterations that seem to have to do with altered gene expression. So anyway, down the road, you've got all that laying before you, right?
Yes, we have a project going on now. A talented postdoc in the lab is doing sequencing of the basolateral amygdala, like bulk sequencing, not single cell, but trying to find differentially expressed
But is
genes.
this looking at RNA-seq?
Yes.
Yeah, that's RNA. Okay, yeah.
So he's actually using this model, the deconstructed adversity, where they're shocked alone, shocked with the mom, they're just placed in a beaker alone for an hour and a half.
So that's like isolation type.
Yeah. And so he's taking all these different little types of stress and he's going to sequence all of those, looking at the basolateral amygdala, looking at sex differences in like differential expression of genes controlled by, sex. And it's interesting that we don't really see sex differences in the behavior, but there are some early biomarkers that are differential, like sexually dimorphic after adversity, which is really interesting. And that those might becomes behaviorally relevant after puberty, for example. Like we've seen, this is just very early pilot data. I don't even know if I should talk about it, but we see, for example, in the endocannabinoid system.
Well, I'm not very good at, I don't have a Facebook account. I don't have any social media accounts. So I need to figure out a way to make more people aware of these podcasts. But I can guarantee you there's nobody listening to this that's going to scoop you.
Oh, no, it's not even that. I just feel like it's irresponsible for me to say
no,
it as
sure.
though it's
Yeah,
like
yeah,
a fact when it's
yeah.
just early days. But we do see that there's some evidence of changes in endocannabinoid signaling in the basolateral amygdala only in females after adversity, like right after adversity, which another student is doing that right now to study, following up on that for her part of her PhD. But you know what's interesting? A lot of people come into the lab and they'll want to study like impulsivity or habit forming, and these are all really interesting behaviors, right? Or constructs, but it's really hard to model them effectively in pups, right? And so sometimes it's like, so I didn't want to keep animals around longer than, the first month, because this is like, there's so much work to be done in the first month, but I caved in and now we're keeping animals all so that they grow up and we can look at, because that is really interesting. It's just, of course. And that's really,
or even the next generation, right?
of course. Yeah. And, we're going to look at some outcome studies and maybe some of these additional measures of behavior and how early stress can remodel circuits to disrupt those behaviors. But in terms of the transgenerational stuff, one really interesting finding that was led by my colleague, Rose Perry, showed that pups, it's not really transgenerational, I guess I would say, but it isn't, it's still within the same generation, but the pups that were abused in early life, or they were maltreated, I guess I would say, when they grow up and they are mothers, like the females grow up and they have their own pups. Normally, when you put this fox odor in a chamber, the mother rats will hide their pups. But these animals that had been adversely reared bring their pups to the fox odor, like to the source of it. Yeah. So it's, again, suggesting that like the processing of what is threatening and safe is totally disturbed.
Wow.
Yeah.
That is crazy.
And yeah, I mean, it's crazy because it's like, this always comes up in conversation, but it's an understudied thing for sure. That would be it. Yeah.
Huh. And
but again, that's another thing, like these adaptations. I mean, that doesn't seem like an adaptive adaptation, but like I said, like the adversity rearing moms, like they'll start making nests out of food. So maybe they have more cognitive flexibility. Maybe there are some advantages to dealing with stress. And I think Bruce Ellis's work, I think he's at the University of Utah, about like hidden talents is sort of an interesting framework for this, because I think a lot of children who grow up with adversity are sort of stigmatized because it's always viewed as like in a deficit model. But if you, I guess what he's arguing is that like changing the testing a little bit can uncover like hidden talents.
oh, interesting.
Which I think also kind of goes with, in my own thinking, like I started by talking about these changes in social behavior in the rats, the dominance hierarchy, how normally they should prefer what happened, which I didn't get into, normally they, given the choice between an animal they've never met and a familiar animal, rats really like social novelty. So they'll prefer to interact with a partner they've never met. But if they've had this disruptive social experience, they'll now prefer to interact with a familiar animal. And that might actually be the right move for them. You know what I mean? Like that's not necessarily like we see that as a deficit, but it could also be viewed, especially since the experience was pretty brief, like it's a remodeling of the circuit, the hippocampus circuit, of the social behavior to suit the environment.
Now, one thing, what if you change the, so you hear a lot about oxytocin, and actually I don't know if you know Larry Young at Emory. So he was on one of the podcasts, and so oxytocin is like, if you Google oxytocin, you'll get like all this, you know, you're gonna be wonderful if you take oxytocin. But so when a mother gives birth, there's a surge of oxytocin, right? And it's thought to play some role in mother-child infant bonding. So what if you take pups that have been born to one mother and put them with a mother that hasn't given birth and hadn't had the oxytocin surges? How does that affect development of the pups?
Well, I don't know that she would be able to nurse them, right? If she.
That's true.
So I think that they might still.
Yeah. Yeah,
I don't know if they would still approach her, because
you have to have your graduate students nurse them with a little bottle of milk.
Exactly. Or just give her oxytocin, you know, But I think, I mean, I think the oxytocin, we know it's really important for the maternal bonding to the infant, but I think there's less known about the role of it in infants. So I think the circuit for attachment in pups is more reliant on norepinephrine and the odor learning system. Like locus coeruleus releases norepinephrine, the olfactory bulb, and they have this really powerful norepinephrine circuit that's promoting odor-based learning towards them, and that starts like in the utero and in utero and after.
So to finish up, so when I introduced you, I mentioned that you're at the Kennedy Krieger Institute. Is your lab in that building? Where's your lab at?
Yes, my building is at Kennedy Krieger. I have an appointment here, and I have my primary appointment, I guess I had primary in both, but it's at the Department of Neuroscience, in the School of Medicine. But my lab is located here, and part of my hiring is this. So Kennedy Krieger Institute's really, focused on the origins of developmental disabilities in children, and I was part of a hiring for basic research, and especially there's a center for the neuroscience of social injustice, and that's kind of the early life stress piece, the resource scarcity piece. Like, how does that change the brain of children, and how do we identify a really important thing to take away, I think, is that you can't just take the adult and use that to answer questions about interventions and therapies for children, because the infant brain and the child's brain is different than the adult, and it's functioning differently. And so we're hoping to target these time points when intervention might be more effective for children, And so I think that's what, that's sort of the synergy of, our work with the clinical questions that are being asked at Kennedy Krieger, try to identify better therapeutics and interventions for children.
So I have an interesting experience with Kennedy Krieger. It was, so my family and I moved here from Kentucky in 2000, and my son Elliot, and this was just a coincidence. So one of his friends at the elementary school he went to in Lexington, Kentucky, his parents moved to Bethesda, to NIH main campus. And about a year after both of us families moved, Elliot's friend got hit by a car and had a really severe traumatic brain injury.
Oh.
And they took him to Kennedy Krieger. He was in a coma for several weeks. And we'd go up there, take Elliot up there to see him like every weekend for a couple months. And, so he came out of the coma, but he couldn't really talk, and he couldn't move well. But his recovery was amazing. So he must have been 10 years old. But over a period of months, he got his speech back, he slurred some, he did a lot of physical rehab, got able, not perfectly, but he can walk normally. He graduated from high school, actually got a college degree. But yeah, so that's the kind of thing that Kennedy Krieger does. So we were pretty impressed with people there.
They do amazing things here for sure, like unbelievable rehabilitation stuff and child motor function and in typical development and with disruption. It's just, it's an incredible place.
Yeah, good. I guess that's it. I have one last question that I jotted down and kind of mentioned it with the mission of Kennedy Krieger. But so what are the implications of your work for helping caregivers optimize the development of social skills in their child?
I would, as much as you can. I guess it's hard to, I don't want to oversimplify anything, since all of my work is in rats, of course. But as I said, what we see is that the best way to help the child is to help the caregiver. And when a stressed caregiver is less able to regulate the child's stress. And so the best thing the caregivers can do is, I guess, try to keep their own stress levels low, if that's possible, and not be too hard on themselves.
And it is hard. It's a difficult situation, particularly of a mom, the newborn kid. So maybe the mom had, before she had the child, she was like exercised all the time and, was able to eat well and so on. And then you have the child and you got to devote all your time, the time's got to be devoted to the child, being with the child. But then that kind of takes away for things like exercise, which is actually really good for relieving stress, and women will suffer, many with postpartum depression, so kind of trying to figure out how to help
Yeah,
help the women have a little more time for themselves so that they can get exercise, that they can have some social interaction and kind of reduce the
100%.
stress. And how do you address that on a society-wide basis? And you talked about disadvantaged people and stuff, and in Baltimore, of course, and like inner city, a lot of places, it's people have kind of tough situations.
Yeah, I mean, I think it would be great if this work could be used to inform policy about resources for caregivers. Again,
But
just
anyway, but what happens is your work, you'll go to a meeting, right, on whatever early life adversity or development of social skills, and there'll be a lot of people that work in humans and some that work in animal models, and you will talk to each other. And hopefully, unfortunately, a lot of times the stumbling block is getting what the scientific knowledge translated into actually something happening. I've done a lot of work on intermittent fasting and how that can improve health and maybe even brain health. I mentioned exercise. How do you or how do you figure out how to free some time of a mom so that she can take care of herself? And that's something beyond anything you and I know how to do. Yeah. But there's kind of a rate limiting step there. And I think there's a big gap between what we actually know at the science level and what policy is. So
that's true. And I mean, it's tough because you don't want to, in some sciences that you don't want to rush
no,
and you also don't, and I also said, like, we don't want to stigmatize people in poverty with this work. And so, but one thing that's been I think is helpful is Regina Sullivan, who was my postdoc mentor. She's really leading the charge to like write papers with human researchers that directly have like cross-species translational like data and perspectives, and like, what are we talking about when we say this age in a rat versus this age in a child? And like that alone is actually quite challenging to like get people to, because these literatures are so siloed, really. And there's a lot of like lip service about collaboration and translation, but it is really hard to actually get people on the same page. It's a lot of work, and it's a lot easier to just stay in your own lane. But that work, I think, is really important, and sort of it increases the impact, I think.
Yeah, I agree. Okay, Maya, I appreciate you taking time out of it's an exciting time for you. I remember when I was, getting going with my lab and bringing in graduate students and postdocs and getting all my equipment in and set up. That's really exciting times. So have fun.
Thank you so much. It's been great chatting with you. Thanks for inviting me.
All right. Bye.
Bye.